There's a question that geneticists have been whispering about in laboratories for decades, a question so loaded, so historically charged, so scientifically explosive, that most mainstream outlets have chosen to dance around it rather than confront it directly. The question is this: who, at the molecular level, are the Jewish people? Not politically, not theologically, not culturally, but biologically, at the deepest level of human existence, inside the strand of code that makes you, you. Because buried inside the DNA of Jewish communities scattered across 5 continents, from the ruins of Eastern Europe to the sunlit markets of Morocco to the mountains of Ethiopia, scientists have found something that should not statistically exist: a thread. Ancient, unbroken, and almost impossible. A thread that has survived 2,000 years of exile, persecution, forced migration, mass murder, and cultural erasure. And that thread, that invisible biological signature, is now being decoded by the most advanced genetic tools humanity has ever built. What they are finding is rewriting history, not adjusting it, not refining it, rewriting it. And most of the world has absolutely no idea this is happening. Let that sit for a moment. Because what comes next is not a religious argument, it is not a political statement, it is science. Cold, peer-reviewed, ancient DNA science that is pulling back the curtain on one of the longest human survival stories ever recorded on this planet. To understand where this story begins, you have to go back, not hundreds of years, but thousands. Roughly 3,000 years ago, in a narrow strip of land along the eastern coast of the Mediterranean Sea, a group of people lived in what scholars call the ancient Levant. This region, encompassing what is today Israel, Palestine, Lebanon, and parts of Jordan and Syria, was one of the most geopolitically turbulent zones on Earth. Empires collided here, armies marched through here, trade routes crossed here, and in the middle of all of it, a tribal confederation called the ancient Israelites built something that no army could conquer and no empire could erase: a cultural and biological identity that would somehow survive everything history had waiting for it. And the proof of that survival is not in a scroll, it is in a cell. Modern genomic analysis has fundamentally reshaped our understanding of the Jewish diaspora by identifying a persistent biological link to the Levant. Despite nearly 2 millennia of geographic separation, diverse Jewish populations, including Ashkenazi, Sephardi, Mizrahi, and North African groups, demonstrate a significant degree of genetic proximity rooted in ancestral markers that trace back to the Iron Age Middle East. Think about what that means. Communities living in Poland, in Yemen, in Spain, in Ethiopia, separated by oceans and centuries, are carrying overlapping pieces of the same ancient biological code. That is not a coincidence. That is a story. And science is now finally fluent enough to read it. But here is where it gets complicated. Because the Jewish people are not, genetically speaking, one single monolithic group. They are a mosaic, a collection of distinct communities that each absorb different genetic influences from the host societies around them, while simultaneously preserving a core of ancient Middle Eastern ancestry that was never fully diluted. The 3 major branches, Ashkenazi Jews of Central and Eastern Europe, Sephardic Jews of the Iberian Peninsula and Mediterranean, and Mizrahi Jews of the Middle East and North Africa, each carry their own layered genetic story. And yet, astonishingly, when you line up their genomes, they cluster closer to each other than they do to the non-Jewish populations they lived among for centuries. That is the paradox at the center of this entire story. Thousands of years of living inside other civilizations, and yet the biological thread was never severed. Some 40 to 50% of Ashkenazi ancestry traces to a Middle Eastern source reflecting the ultimate origins of the Jewish population in the ancient Near East and the Levantine diaspora of the Second Temple period. About 50 to 60% traces to a European source, primarily Southern European, consistent with the admixture that occurred as Jewish communities interacted with the populations of the Roman Empire across the first millennium of the Common Era. So the Ashkenazi genome is not purely Middle Eastern, nor is it simply European. It is both, a biological hybrid forged by centuries of migration, interaction, and isolation. A mosaic within a mosaic. But that dual ancestry is only the beginning. The more you look at the Ashkenazi genome, the more a single event starts to dominate the entire picture. An event so severe, so genetically defining, that it compressed the biological diversity of an entire civilization into a single tight cluster. Scientists call it the bottleneck, and understanding it is the key to understanding almost everything else about Ashkenazi genetics. Modern genome sequencing suggests that the Ashkenazi population underwent a severe contraction in size approximately 600 to 800 years ago. At its lowest point, the ancestral population may have consisted of only a few hundred individuals. This bottleneck led to a powerful founder effect, where the genetic traits of this small group became the blueprint for millions of descendants. Read that again. A few hundred people. From a few hundred people emerged a population that today numbers in the millions. And because of that catastrophic compression, every single Ashkenazi Jew alive today is genetically descended from that tiny group of survivors, which means that if you are of Ashkenazi descent, you and any other Ashkenazi person on the planet, whether from New York or Tel Aviv or Buenos Aires, are, by genetic standards, distant cousins. That is not metaphor. That is documented genomic reality. The Ashkenazi population descended from a relatively small founding population that experienced a severe bottleneck, possibly as few as 350 individuals, in medieval Europe, likely around the 13th to 14th century. This small group then expanded rapidly, but because they married primarily within the community, their genetic diversity remained limited. The endogamy, the practice of marrying within the community, was not merely a religious tradition, it became, unintentionally, a mechanism of genetic preservation. Every generation that chose to marry within the group reinforced the same core of ancestral markers. And what you get, after centuries of this pattern, is a population whose genome is so distinctive that researchers working on genome-wide studies in 2009 at Duke University could computationally separate individuals with full Jewish ancestry from those without it with near-perfect accuracy, across a random sample of thousands of people, not by asking them, not by looking at them, by reading their DNA alone. And then the ancient DNA evidence arrived. And it confirmed everything. Genome-wide data from 33 Ashkenazi Jews, dated to the 14th century, obtained from a salvage excavation at the medieval Jewish cemetery of Erfurt, Germany, showed that the Erfurt individuals were genetically similar to modern Ashkenazi Jews, but displayed more variability in Eastern European-related ancestry. 8 carried pathogenic variants known to affect Ashkenazi Jews today. These observations, together with high levels of runs of homozygosity, suggest that the Erfurt community had already experienced the major reduction in size that affected modern Ashkenazi Jews. Scientists dug up 14-century-old teeth from a Jewish cemetery in medieval Germany, extracted DNA that had survived 700 years underground, and found that those individuals genetically resembled the Ashkenazi Jews of today. The bottleneck had already happened. The founder effect was already locked in, and the biological signature that defines an entire people was already written, centuries before the worst chapters of their persecution even began. But here is where it gets even more layered, because the bottleneck is only one dimension of the Ashkenazi story. Equally fascinating, and considerably more complex, is the question of where the maternal and paternal lineages actually came from. Because when you separate the Y chromosome data from the mitochondrial DNA data, something surprising emerges. The fathers and the mothers are not telling the same story. Paternal lineages tend to trace to the Middle East, while maternal lineages show strong European roots, reflecting complex migration and adoption patterns. The men, in other words, carried genetics pointing back to the ancient Levant. The women showed a stronger European signal. This asymmetry is not random. It suggests that when Jewish communities first established themselves in Europe, Jewish men, traders, merchants, scholars, may have settled in new territories and married or formed families with local women, who then converted and raised their children within the Jewish tradition. Over generations, those maternal European lineages became incorporated into the community's genetic profile, while the paternal Levantine lineage was preserved through patrilineal religious inheritance. This is population genetics intersecting with social history in real time. And informed sources in the field will tell you that this particular asymmetry has been one of the most debated findings in the entire discipline of Jewish genetic research. Some researchers argue the maternal European signal is stronger than expected. Others point to a 2025 study in Science Direct, which concluded that both maternal and paternal lineages ultimately share a Near Eastern ancestral origin, with European maternal lineages representing absorbed rather than foundational genetics. The debate continues, but the core finding, that Ashkenazi Jews carry a genuine ancient Middle Eastern genetic signature alongside European admixture, is not in dispute. That part is settled science. Now shift your gaze from the Ashkenazi world to something even older, something that predates the European migrations, predates the bottleneck, predates nearly everything. The Y chromosome of the Jewish priestly caste, the Kohanim. In 1997, geneticist Karl Skorecki made a discovery that sent shockwaves through both the scientific community and Jewish religious circles simultaneously. He discovered that a disproportionate number of Jewish men with the surname Cohen, or variants like Cohn, Kahn, Cohen, share a distinctive Y chromosome signature, the Cohen modal haplotype. Since the priestly status is passed from father to son, this genetic marker suggests a common patrilineal ancestor, possibly dating back roughly 3,000 years. Think about the implications of that. A religious tradition, passed down through oral history and scripture, designating a specific group of men as the descendants of Aaron, the brother of Moses, the first high priest, was suddenly corroborated by molecular biology. The DNA matched the story, not perfectly, not universally, but statistically, undeniably, in a way that cannot be dismissed. Some 46% of all Kohanim carry on their Y chromosomes a specific genetic signature, one that does not appear to exist in non-Jews. The first bearer of this signature is estimated by genetic calculation to have lived some 300 years ago, a date that fits with archaeological evidence for the beginning of Judaism. 3,100 years ago. That is not the medieval period. That is not the Roman Empire. That is the Bronze Age. And embedded in the Y chromosomes of Jewish men who self-identify as priests today, men living in Los Angeles, in Jerusalem, in London, in Buenos Aires, is a biological timestamp from that ancient world. The priesthood was not just preserved culturally, it was preserved genetically. And yet, as always in this story, there's a complication that forces you to look deeper. Because the Levites, another traditional tribe within Jewish heritage, considered the priestly assistants, tell a different genetic tale entirely. Unlike the Kohanim, Ashkenazi Levites show evidence of multiple paternal origins. Over half of Ashkenazi Levite Y chromosomes belong to a haplogroup called R1A1, which is common in Eastern European populations, but rare in Sephardic Levites or other Jewish groups. This suggests that at some point in history, European males entered the Ashkenazi Levite lineage, possibly through conversion or intermarriage, creating a genetic pattern unique to this specific subgroup. The Levites, in other words, absorbed outside genetic material in a way the Kohanim did not, or at least did not to the same degree. Two tribes, two completely different genetic trajectories, both inside the same community. This is what makes Jewish genetic history so layered, so resistant to simplification. It is never a single clean narrative. It is always multiple overlapping stories operating simultaneously. Now we come to perhaps the most medically consequential dimension of this entire genetic saga. Because the bottleneck that shaped Ashkenazi Jewish ancestry did not only preserve ancient lineages, it also concentrated specific genetic mutations, and some of those mutations carry serious medical consequences for people alive today. The high prevalence of some 20 Ashkenazi diseases dates from founder effects and bottlenecks in the era after 75 CE and between 1100 and 1400 CE. The most common are the neurodegenerative Tay-Sachs disease and Gaucher type 1 disease, which result from enzyme deficiencies. Tay-Sachs is a devastating condition. It destroys nerve cells in the brain and spinal cord, and in its most severe form, children born with it rarely survive past age 5. And because of the founder effect, because the genetic blueprint of the entire modern Ashkenazi population traces back to that small medieval group, the mutation responsible for Tay-Sachs became far more concentrated in Ashkenazi communities than in the general population. About 1 in 30 Ashkenazi Jews carries a Tay-Sachs mutation, 1 in 30. Contrast that with the general population, where the carrier rate is roughly 1 in 300. That is a tenfold difference, written directly into the genetic code by a historical catastrophe that occurred 7 centuries ago. And then there is the BRCA story, perhaps the most consequential genetic legacy of the bottleneck for living people today. Ashkenazi Jewish women and men have approximately a 1 in 40 chance of carrying a BRCA mutation, compared to 1 in 500 in the general population. These mutations increase the risk of breast, ovarian, pancreatic, prostate, and other cancers. 1 in 40 versus 1 in 500. That is not a marginal statistical variation, that is a gulf. And for years, many people in Ashkenazi communities did not know this number. They were walking through life carrying a genetic time bomb inherited from a founder population that lived in medieval Germany, without any awareness that the risk was even elevated. The science was there. The awareness was not catching up fast enough. That is changing. The NHS England Jewish BRCA testing program is offering germline BRCA1 and BRCA2 genetic testing to people with at least one Jewish grandparent who have an increased likelihood of having an Ashkenazi Jewish founder pathogenic variant compared with the general population. The United Kingdom, for the first time, launched a national program specifically targeted at this population. It is a historic moment, one of the first times a government has built a healthcare pathway explicitly designed around the genetic history of a specific ethnic and religious community. And the results in the program's first year were striking. Thousands of people self-referred. Hundreds of carriers were identified who would otherwise have had no idea. And behind each of those positive results is a family, a mother, a daughter, a sister, who now has information that could change their medical decisions, their surveillance protocols, their very survival odds. But we are not done with the geographic spread of this story. Because the Jewish genetic narrative is not limited to Europe and the Middle East, it extends into regions that would astonish most people who have never looked at this data carefully. Consider the Lemba people of southern Africa, when researchers found that some of the Lemba carried the Cohen modal haplotype, a set of genetic markers found among some Jewish men who identify as Cohens, this was considered evidence that they were likely descended from Jews in ancient Israel, consistent with their oral history. The Lemba have long maintained an oral tradition claiming descent from ancient Israelites. For generations, scholars filed this claim under folklore. Then the genetics arrived, and the Cohen modal haplotype showed up in their Y chromosomes. Suddenly, the folklore was not folklore anymore, it was a lead, a biological lead pointing back to the ancient Levant, all the way from the southern tip of Africa. Now we arrive at the question that hangs over all of this, the question that sits at the intersection of genetics, identity, history, and geopolitics in a way that no simple answer can resolve. In a study of 1,171 men from around the world, geneticist Michael Hammer of the University of Arizona found that the Y chromosome in Middle Eastern Arabs was almost indistinguishable from that of Jews. Almost indistinguishable. Let that land. The two peoples most associated with one of history's most intractable conflicts, a conflict framed so often in terms of fundamental difference, irreconcilable opposition, existential incompatibility, share Y chromosomes so similar that they cluster in the same genetic haplogroups. According to informed sources in the field, the genetic distance between Jewish and Palestinian Arab populations is, in many measures, smaller than the distance between different Jewish subgroups. The Arabs and the Jews of the modern Middle East are, by the measure of molecular biology, cousins. Not metaphorical cousins, genetic cousins, sharing common ancestors within the past several thousand years. This is not a political statement. This is not an argument for or against any territorial claim or policy position. It is simply what the data shows. And the fact that this finding is not common knowledge, the fact that the image of two ancient enemy peoples has been so thoroughly constructed around their differences rather than their biological proximity, is itself a statement about how selective our relationship with scientific evidence can be when politics is in the room. What the real story reveals is that the concept of total genetic separation between these communities is a fiction. The ancient Levant produced both of them. The same land, the same Bronze Age ancestors, the same haplogroups, diverging over centuries of separate history, war, religion, and forced separation, but never fully severing the biological connection at the root. Over the past 3,000 years, both the flow of genes and the flow of religious and cultural ideas have contributed to what it means to be Jewish, with genetic threads observed as identical strands of DNA shared within and between Jewish groups across the world. 3,000 years. That is the time frame scientists are now working with. Not hundreds of years, thousands. And across that span, through every empire that rose and fell around them, through every expulsion and inquisition and pogrom and genocide, the Jewish people carried something that could not be confiscated or burned or erased. They carried it in their cells. And here is what that means in the context of the most advanced genetic research happening right now, in 2024 and 2025. Ancient DNA technology has reached a point where scientists can extract genomic data from a 1,000-year-old tooth and run it against modern population databases to determine ancestry with extraordinary precision. By integrating 2024 to 2025 benchmarks from the Ashkelon time transect, examining Iron Age Levantine inhabitants from roughly 1200 to 586 BCE, researchers have demonstrated that while the Palestinian genomic profile represents high continuity derivation of the regional gene pool, the Ashkenazi profile reflects a permanent sex-biased admixture event that distinguishes it from the regional baseline. This is not old science. This is not Cold War-era blood typing. This is high-coverage ancient DNA analysis, 161 studies screened in a single systematic review, using the most sophisticated statistical modeling tools in population genetics. And still, the core signal holds. The Middle Eastern ancestry is there. The Levantine thread is there. It did not disappear. It adapted. It absorbed new material, but it never vanished. This is the story that most people are not seeing when they look at Jewish history through the lens of politics or religion alone. They are missing the biological dimension, the molecular testimony of a population that somehow encoded its own survival into the most durable archive that exists. Not stone, not papyrus, not digital data, which can be corrupted in a server room. DNA, the four-letter code that has persisted through ice ages and mass extinctions. The Jewish people wrote their history there involuntarily, without intention, simply by surviving. And now, for the first time in human history, we have the tools to read what they wrote. What does this all mean going forward? As genetic testing becomes cheaper, more accessible, and more precise, as companies like the NHS build targeted programs, as ancient DNA studies expand to cover more burial sites and more time periods, as whole genome sequencing becomes a standard clinical tool, the portrait we are drawing of this population is going to become more detailed, more nuanced, and almost certainly more surprising. Every new excavation is a potential revelation. Every new genome sequenced from a medieval Jewish cemetery in Germany or France or Spain could shift the current understanding of migration patterns, bottleneck timing, or the origins of specific disease mutations. And the questions that remain unanswered are enormous. We still do not fully understand why the bottleneck occurred when it did. Was it the Black Death? Was it the mass expulsions and massacres of Jewish communities across Europe in the 13th and 14th centuries? Was it a combination of both? What happened to the branches of the Jewish world that did not survive? The communities wiped out before they could contribute their genetics to the modern pool. How much of what we call the Ashkenazi genome today is actually the genome of those who were fortunate enough to escape, while others left no descendants? How different might the genetic picture look if history had been even slightly less violent? And there is a deeper question still, one that science can approach but not fully resolve. What does it mean for people's identity to have a genetic dimension? The Jewish people have been defined for millennia by religion, by law, by language, by memory, by shared trauma, and shared aspiration. Now there's a molecular layer on top of all of that, a layer that crosses denominational lines, that connects communities who have never met, that links the living to the ancient dead in a way no scripture or ceremony can replicate. It does not simplify the question of who is Jewish. It vastly complicates it. And it forces a reckoning with the fact that identity, human identity, all of it, is never just one thing. It is always a palimpsest. Layer upon layer, each one inscribed over the last, none of them fully erasing what came before. The DNA of the Jewish people is not the answer to that question, but it is one of the most powerful witnesses to it. A witness that has been silent for 3,000 years and is only now finally beginning to speak. The question is, are we ready to listen to what it is telling us, and what else is written in that code that we have not yet found the language to read?